Okay, what are disulfide bonds formed by? Okay, so it's basically if you were to draw like a sulfur here connected to another sulfur, right? That's what that is. Good, that's a disulfide bond. It's formed by, in this case, the oxidation and reduction, in this case oxidation, because we're taking a hydrogen out, of cysteines. And so if you have a cysteine amino acid, let's say you had a bunch of cysteine amino acids, that would tell you, okay, this probably makes something that's either globular or something that's filamentous. You don't need to know the identities of any amino acids other than cysteine for the purposes of this class. So that's like the main thing, because covalent bonds are really, really strong, relatively speaking, right? And that holds together things pretty gosh darn well. Ever gotten a perm? Have you ever had someone who lit your hair on fire? Really? Did you not live? No, I'm kidding. If you ever had to smell burnt hair, what does it smell like? Does it smell good? No. But if I'm burning a hamburger, that smells good, relatively speaking. Burnt hair, rotten flesh, all that stuff smells terrible because what you're doing is you're breaking these bonds and you're releasing sulfur in the air, and sulfur does not smell good. Okay, so that's disulfide bonds. The other thing we're just going to say is collectively non-covalent forces. What are some examples of non-covalent forces? Anything that's not, yeah. So van der Waals, ionic interactions, hydrogen bonding, all of that stuff is there. Oh, you don't, I mean, yeah, you can write that. Yeah, go ahead. Okay, good. So that's pretty much it for tertiary. Okay, so what is the quaternary? If this is the final 3D structure of the protein, what is quaternary?
It's the final. That's because some of them skip secondary, tertiary, and tertiary.
Oh, okay. I'm glad that you're saying that. So yeah, it's not like this is a linear process. A primary protein doesn't become a secondary protein, and a secondary protein doesn't necessarily become a tertiary protein. Not necessarily. You can have some proteins that will form these very weird kind of secondary structures, and they'll just stay secondary, and they won't do anything past that. And then you have some that form these weird ball-like shapes that I wouldn't even say are. It's just this. I mean, there will be alpha helices and beta sheets in it, but it's not alpha helical, if that makes sense. The subunits of it. Quaternary, and this is how, like I said, this is an arbitrary man-made classification system, so it depends on who you ask. Quaternary level protein structure is where you have more than one polypeptide chain coming together to form a protein. So just write more than one polypeptide chain. Okay, good. I don't know why I have a very weird collection of like random facts in my brain. The most abundant protein in the human body is called collagen. Do you know what collagen is?
It's in your hair. Oh, I can do
It's a part of connective tissue. The reason that your bones can bend without breaking, which is really what makes them so good at resisting so much PSI, is because they have collagen in them. Collagen is multiple protein subunits. So your body makes one protein, makes another protein, and then a third protein. And those three proteins, all three of them, come together to make this one super protein called collagen. And it is. Is it more than one polypeptide chain? Yes. Hemoglobin. Do you know what hemoglobin is? It's in your blood. Hemoglobin consists of 1, 2, 3, 4 globular subunits, all four of them coming together and interacting. Is that quaternary?
Yes.
Yeah. Is it more than one polypeptide chain? Yeah. It's 3 globular subunits, all 3 of them coming together to give us that. So quaternary is actually held in place by, well, non-covalent forces as well. So you can just draw an arrow down there connecting those two. All right, good job. Do you want to take a picture of this before we fill in the blanks? All right. All right. You got this. Good. Hydrogen bonding where? Between the R groups or between the backbone?
Backbone.
Good, backbone. So, good. Let me think. I'm trying to think of questions to ask. That's pretty much it. I could divulge a bit more in beta sheets on parallel versus anti-parallel, but let's move on to nucleic acids. That's the last biomolecule I think we need to talk about. May I look at your notes real quick just to make sure we're staying on track?
Yeah.
Where are they? Okay, I see them. Okay, good. Okay, so then he goes into enzymes.
Yes.
Okay, let's talk about enzymes next then.
We didn't really touch on
Nucleic acids. I think he'll talk about that whenever he mentions like DNA replication and other things. All right, so what can you tell me about enzymes? What are they? What do they do? Why do we make them? All that stuff. Structure and function.
Really help us structure and to
Well, let's start off. So biologists are really kind of pricks about classification of things in terms of like having very strict, and I get in trouble with this all the time, very strict definitions about things. I'm always quick to say what's in a name, and so long as someone understands the concept, but it's really important that biologists be able to communicate with each other so we're all speaking the same language. So, what is the definition of an enzyme?
well, it's like part of proteins and it's a macromolecule.
Good, but not all enzymes are proteins. And what are they though? Like, I know we're saying that they're proteins, but what is their functional role? I know I asked very ambiguous, open-ended questions. There's,
I can't tell you that right now.
that's okay. Okay, enzymes are biological catalysts. So, underneath enzyme, I want you to write just like biological catalysts. Okay, so biological means obviously there's some type of a biomolecule. Two biomolecules that make up enzymes, do you know what they are? They can be enzymes.
Protein.
Proteins, and what else? Ribonucleic acids. Those two things can act as enzymes. What is a catalyst? So you were chemi and you're like, I'm gonna do biology or biochem, right? Okay, what do catalysts do? Catalysts speed up the reaction. But are they consumed by the reaction? No, catalysts are not consumed by reactions. They speed them up. How do they speed them up? They lower the energy barrier, the activation energy barrier. Catalysts, whether you're using palladium on carbon or you're using chymotrypsin, both of those guys are going to lower the activation energy required for that reaction and enable it to go faster. In this context though, we're saying the unique property of enzymes is that enzymes are super specific catalysts. If I use, do you remember from ochem palladium on carbon by any chance?
Mm-hmm.
What does that do?
It's an active catalyst, and we should form an ochem2 bond when we study it for hydrogenation.
Yeah. It's a really good reduction. Yeah, and it doesn't care. It doesn't care whether you put in octane or, octene or whatever. Anytime it sees a double bond, it's gonna immediately reduce it. It doesn't care. Enzymes are not that way. Enzymes are really, really, really picky about what it is, the types of reactions that they catalyze. So the way that we're gonna break this down is obviously based off of their structure and off of their function. So biological, we'll just go ahead and write out the two biomolecules that are use this. So just draw two arrows underneath biological. Good. Go ahead and write RNA up here, because that's not talked about a lot for some reason. I guess because it's a fairly recent discovery, and underneath that, protein. Okay, I'm so sorry. I'm not being helpful. Right, protein in there. Good deal. Okay. If it's a protein, we call it, what is the nomenclature actually? We didn't discuss this. What is the nomenclature of an enzyme? If I say carboxyl peptidase, you automatically know that that's an enzyme because it ends with what? Ase. Good. So carboxypeptidase, acetylcholinesterase, anything that ends with ase means it's some type of an enzyme. Okay. Catalysts. What do we say the mechanism was of catalysis for these guys? Speeds up the reaction. Does it make the reaction happen? Does it make an impossible reaction possible?
Mm-hmm.
No. It doesn't make an impossible reaction possible. It just speeds up the reaction that's going to happen. How does it speed up the reaction? In terms of physical chemistry, how does it speed up the reaction? There's a lot of mechanistic strategies that they have, but it lowers the energy barrier, lowers the activation energy, the EA. What is activation energy actually? Let's talk about that. So go ahead and just draw me a graph with an X and Y. Okay, and on the y-axis, just say E for energy. And then for the x-axis, we'll just say reaction. We would say reaction coordinate, but we're not bricks. Okay, so, sorry. So I want you to draw a line here for the reactants, an energy barrier, and then another line down there for products. Perfect, beautiful. Okay, so, what is your chem background? Tell me about that a little bit. Gen Chem. Okay, my experience at Tech, Gen Chem is like Russian roulette, but with like 4 rounds in the chamber in terms of the professor that you get. Really bad, like it's a punishment. If someone teaches Gen Chem at Texas Tech, the chem department's basically punishing them. It's not like this, like he teaches this bio class every semester, and that's all that he does. The guys who teach Gen Chem, one semester they'll be teaching Gen Chem, and the next they'll be doing, I don't know, X-ray crystallography or something like that. So it's really, really difficult in that you don't get a consistency with your instructors. Changes in, let's just say changes in enthalpy, but also changes in Gibbs free energy can tell us whether or not a reaction is what we would say spontaneous or non-spontaneous, right? Based on whether or not we have to put energy into or energy out of that system. Is this reaction, just by looking at it, is energy going into the reactants, into the chemicals, or is it leaving the chemicals? Good, it's being released from the chemicals. So is this a thermodynamically favorable reaction? Yeah, yeah, so the delta G, delta G, the change in Gibbs free energy with this would be negative, right? If we took the final 3 minus the initial 10, 3 minus 10 is not 7, 3 minus 10 is negative 7, right? So we'd say that it has a negative change in Gibbs free energy. This is energetically favorable. We want this to happen. This will release energy when we do that. To give you an example, let's say I take a steak. Is there potential chemical energy in a steak? Yeah, right? That's why we eat it. But if I just sit it in a cup of water, what's it going to do? It's just going to sit there. Even though it's thermodynamically favorable for that steak to break apart, it's not going to dissolve in the water. It's going to just sit there. But if we add an enzyme and it lowers that activation energy, it starts to break up. Do you know what an enzyme we use? Papain, papain. That's something that you use to break up, to tenderize the meat, because it literally starts breaking it up. Okay, good. I'm glad that you get that. So lowering the activation energy barrier. Does this change the thermodynamics of the reaction? No. It doesn't change the energy states of the reactants and the products. It just makes the reaction happen faster. It doesn't change the thermodynamics of it. It just makes the reaction happen faster. I'm really stressing that because he's going to ask you a question on it, I can guarantee. Okay, let's see what else we should mention about enzymes. Okay, so he goes into some of the mechanism for it and how that happens. It's a good time probably now to mention something for this called a conformational change. So draw an arrow down there and let's just talk about what that is. Okay, so can I see one of those markers? I'm going to draw something. I'm not good at this, and I shouldn't be drawing because I'm bad for my drawings, but there's really just no other way. So let's say that I want you to draw this line. This is an amino acid sequence in the protein. So you have this, voila. Tertiary level protein structure, hydrogen bonding, disulfide bridges, all of that stuff is there. Hydrogen bonds, disulfide bridges, all that stuff, okay? Let's say that this is, I don't know, we'll say this is sucrase. So sucrase is the enzyme that catalyzes the breakage of sucrose. Sucrose is table sugar, and it's a combination of glucose and fructose. So and glucose and fructose, sugars, are they polar? Does sugar dissolve in water?
Yes.
Yes. Okay. So there is this region in the enzyme called an active site. What happens when a sugar molecule binds to that active site? Is it polar? The sugar is polar.
They
So-
look, they look, he does like this and then it comes out.
Good, good.
And then this goes.
Ooh, so good. I'm so glad you get that. What is that called?
This.
Good. Conformational change. So what happens is, like I said, three-dimensional structure.
It's around it.
Yeah, yeah. So it's, you're getting it. It's a three-dimensional structure. It's totally dependent upon the interactions of hydrogen bonding between certain specific groups. And whenever a sugar molecule, or anything really, that it's compatible with bumps into it, that molecule contains, well, either polar or even non-polar molecules that are going to undergo non-covalent interactions. So when the substrate, in this case sucrose, binds, the whole enzyme changes its shape. It has breaking and reforming of the non-covalent forces. So let's say you have a hydrogen bond here in one state, it gets broken up, and then it goes and interacts with two other ones, and the whole molecule changes its shape. And I hate your textbooks because your textbooks will show it like this. Well, it starts off like this, and then afterwards it's like that. It's like that's not really what's happening. What's happening is you're changing the hydrogen bonding and the salt bridges between the two, and the whole molecule.
There's something today that he was like, your textbook is completely wrong about it.
Yeah, no, it happens all the time. I taught at a high school. My high school, okay, that I taught at, the textbook said that a theory or a hypothesis becomes a theory, and a theory becomes a law. No, right? That's not how it works. Textbooks are they're, I don't know who writes them. You'll have scientists write them, and then other like people who are like instructional technologists or education people come in, and then they'll modify it, and it's bad. Anyways, so when we say a change in shape, that's what we mean whenever that stuff is happening. The concept of the conformational change is super duper important. It's an energy thing. It's chemical reactions are happening. So what happens as this is doing that is it's actually, so say that you have, I don't know, hold on, let's do a diagram example. So you hold on to this, hold on to it like that. Okay, so my hand is the enzyme. These are the active sites, and it stays in place. And it stays in this weird conformation like this for the most of the time, okay? Because this is the most thermodynamically stable position for it to be in. What happens is, let's say that you have a lot of sugar. Well, it's going to bounce around. No. But sometimes, if it bounces in at just the right orientation, these two active sites will come in and they will bind to that molecule. Now, when that molecule binds, that protein's going to do what? Change its shape, conformational change. And sometimes that will even physically put pressure on that bond, but also chemically put some type of a stress on the bond. Let's say that I have a covalent bond here between these two, or can you see where my fingers are? Say that's a covalent bond. Let's say that I had, I don't know, positively charged groups where my thumbs are. Positive charge, electron dense covalent bond. What do you think? Does that stress it? Yes, it stresses it. And then physical stress as well. So that when a water molecule bumps in and hits it right there, it's just very quickly breaks. That's called hydrolysis. But what I want you to really understand is that this environment, the active site, is a perfect environment for this reaction to happen. And that's what makes these things so unique. Sucrose is only broken apart by sucrase. There are other molecules called maltose that this doesn't do anything to. Why do you think that's so awesome? As opposed to like palladium on carbon, where you have to, anytime you see an alkene, it's gonna hydrolyze it, right? Or not hydrolyze it, it's gonna reduce it. But this is very, very specific. And so each enzyme catalyzes a very specific type of chemical reaction based off of the active sites. So conformational changes, super important that you understand that. And then a water molecule can come in and just break it up. Once it breaks, let's go back to that actually. What is it? Was it these two? Once it breaks, okay, I had my thumbs like this. Once these break here, are these, now that they're separate, do they have the same chemical properties as being together? No, we just broke a covalent bond and added water to it. So now that it's different, the moment it bumps into something inside the cell, the substrates leave. I'll give those back to you. And when they leave, guess what? It goes right back to whatever state it was, whatever shape it was like at the beginning of this. So you can actually plot the changes in the enzyme's energy as it undergoes this reaction as well. Okay, super important that you get that. Kind of hard to concept. Now let's just go ahead and conformational change. We got that written down. There's other structural features that we want to make about this. So if we're able to do conformational change, you said it speeds up the reaction. For protein, let's just go ahead and write out tertiary protein. Tertiary protein. So there's two parts that we mentioned. The active site, and then we'll talk about something else later. But for starters, we'll mention the active site. And what is the active site?
Where things come.
Yeah, where the substrate, and okay, that's another thing I didn't tell you this. So in chemistry, we say reactants and products. They say substrates and products. I don't know what it's irritating. It's an arbitrary thing. In this case, substrate is a bit more specific than saying reactants, but who cares? But the active site is where substrates are going to bind and where catalysis happens. So just draw two arrows underneath there. So because this is a tertiary protein, go ahead and just draw that that's the thing with conformational changes. And it's not just enzymes that do that. This feels cold. I can feel cold with my finger. I can feel the texture with my finger. Why? Isn't that weird? Because there's little proteins on the tip of my thumb that are sensitive to temperature or pressure, and whenever the temperature or pressure varies, they undergo a conformational change, and that simple change in shape undergoes a chemical reaction inside their cells that excites the neurons that send a signal all the way up to my brain that says this feels cold, or this feels rough, or this feels whatever. Okay? So it's not just chemical things that can induce a conformational change. Pressure, osmolality, pH, other things like that can induce a conformational change. Your entire mouth, for example, is a series of conformational changes whenever you're eating food. Like peppers, I don't know if you cook. I'm not really good at cooking, but I know that spices tend to activate pathways that make your tongue and the taste buds in your tongue more sensitive because, again, conformational changes. Okay, we get that. We get the concept of conformational change. I'll stop talking about that. All right, so the thing that I want to mention because of this conformational change, it is environmental dependent. So what are some things that affect that conformation?
If it's environmentally dependent, then temperature.
Good. So temperature is one.
Like the aqueous solution around it, would that be one?
Yeah, but we assume, well, yes, but we're going to make the assumption that because this is biology that we don't, I mean, we're not using DMS, well, we do use DMS, so we don't use any like non-aqueous solvents in this context. But yes, that is something. The solvent, what properties does the solvent have can certainly affect the conformation. What else?
Well, like inside of cells, you can't necessarily be like a lighting and stuff in there.
Well, yeah, but there are, like our eyes are sensitive to light. So I love that you're thinking that way, by the way, because that can certainly induce a conformational change. Have you volunteered in healthcare by any chance? Have you ever gone to the thing where they put that little thing on the person's finger and it tells them the level of oxygen saturation? You're literally measuring the conformational changes in the hemoglobin because it gives off light whenever it does that. A certain type of protein called green fluorescent protein, which you may have seen, may have worked with, it doesn't matter. Anyways, it gives off light as conformational changes undergo. And that's a very useful tool. So I like that you're thinking that. What else? But we're not going to write that because that's not like a universally a thing. You know what I mean? The cells inside of your muscles aren't really exposed to light too much. What pH?
Yes.
What about, so pickles are a good example of this. Do you know what pickles are made from? And what else?
Cucumbers.
Cucumbers. So if I take a cucumber, it's a very rigid, very strong, sturdy thing. I dip it in vinegar, which vinegar is what? What is vinegar? An acid. So you change the pH and it does that. What else do you do though? You add
Salt.
salt. So osmolality can certainly affect that. Actually, at my seminar, we were talking about honey, using honey as a disinfectant, or not as a disinfectant, but necessarily for wound care. Yeah, there's botulism toxin in honey, and I always knew that. Not like commercial honey, not like store honey, but like real honey, like raw, yeah, unpasteurized honey.
They sell raw honey.
They do? Where?
Find organic, I think.
Oh,
So
I need-
probably at Market Street.
I need to look into that then. 'Cause anyways,
There's raw honey.
the way that it works is it's really good at controlling infection. Well, it's not great, but it's cheap way to control infection is by creating an osmolality, a certain environment where the bacteria, the proteins inside of them can't really function at an optimal range because you're changing the relative ratio of salt. And what's nice about it is, as you notice, it's really thick and it's
Yeah.
really hard to get any type of diffusion or osmosis going around, so it can relatively, for a long period of time, maintain that high concentration of salt, which is kind of nice. Okay.
So like if you had like a cut, you can put like raw honey on it.
That's what I've read. It's kind of, you need to kind of modify it a little bit because it would have like botulism toxin. I don't know if you know this, but if you ever have a kid, do not give an infant or toddler honey, because like we have an immune system that's totally adjusted and developed, but if you
They
give it to
don't.
a kid, they will stop breathing. It has botulism toxin in it. Yeah. Yeah,
She's like obsessed with honey.
it's actually pretty good for you. There's actually, what's interesting is a lot of the natural or like 1500 wives' tale cures are actually starting to be investigated. Pretty, yeah, you know, we're so desperate at this point, we're like, hey, let's see if that had some validity to it, and some of them do actually. It's kind of interesting. Okay, but does this make sense? Do you understand that if all three of these things here can affect the orientation of hydrogen bonding and non-covalent forces? So it makes sense that if you mess with them, you will have that. Okay, and for each and every one of these things, there is an optimum range for that. Temperature. So if we were to, let's look at our notes real quick.
Our body temperature.
Mhm.
It's like, there's charts on it, 'cause I remember.
Good, okay, yeah. So let's just look at these. You don't have to draw them, but let's just look at these real quick. So let's say that we have an enzyme that, it doesn't have to be an enzyme, it could be, well, yeah, we'll say an enzyme. We'll say carbonic anhydrase, which catalyzes certain types of reactions. It is most effective at that pH there. Not all enzymes do that though. What happens if we have a pH that's super low like this? What does that do to it?
Well, it would shift the graph and mess it up, but they'd have to adapt.
Yes, so what we say is that, so that pH of 7 is what we'd say the natural state for that enzyme. Anytime you take something that's in a natural conformation and you take it out of its natural conformation, we say that that's denaturing it. And that is irreversible. You cannot un-denature a denatured protein. I mean, someone did, but they got a lot of money from patenting that methodology. It's useful for like making drugs and stuff, but for the most part, right? If you know anybody who's ever had like, I hope you don't, but they have like a really severe fever and they had permanent brain damage from it, that brain damage is irreversible. You crack an egg, you heat it, you can't unboil a boiled egg. You can chemically, but not in terms of
Like a normal human being.
Of a human being, yeah, you can. That process of denaturing is really, really, really important, and that's how your immune system works. A lot of times, it will denature the enzymes and proteins inside the bacteria. Okay, so they have an optimal range for any one of those factors that we listed. They have an optimal temperature, right? They have an optimal pH, and they have an optimal salt concentration. And that's really, really, really important. Now, does he mention, okay, he does, good. Ooh, good for him. Good for him. So let's go ahead and go into this real quick. So the last thing that we're going to say about enzymes, and we're just going to draw two big arrows, kind of getting ugly here, but that's okay. Okay, is cofactors. And then on the other side, you're going to write inhibitors. I suppose I could have done a better job with this math, but you know what? You can make it a bit more straight down whenever you do this with yourself. Okay, so let me mention what a cofactor is real quick. Cofactors are some type of a molecule, either a metal or an organic compound, that goes into that active site and makes the active site more conducive to what it is. Do you take a multivitamin? No, you shouldn't. But let's just say, for the sake of this, and I'll explain why in a sec, you shouldn't. It doesn't actually, not a lot of that stuff gets absorbed. Yeah. Linus Pauling is one of the greatest biochemists in history. And he thought, oh, okay, if vitamin C is a cofactor for an antioxidant reaction, then if I just took enough vitamin C, I could live forever. There's a physiological limit to how much of that you can actually absorb. So it's kind of a waste of money to take it in high quantities. So cofactors, we would say, are external substances that make the active site more conducive. Okay, the two types of cofactors that we can have are metals and then organic compounds. Okay, scurvy, do you know what that is? Pirate disease. Rickets, also pirate disease. All the good pirate diseases. Anemia. Anyways, these are what you would usually see on the back of a multivitamin or multi-mineral supplement. So the metals obviously tend to be, well, metals, zinc, iron, whatever, calcium. It doesn't matter. You don't necessarily have to write that down. I don't think he's going to ask it of you. Organic compounds tend to be vitamins. Vitamin B vitamins are used for that. Vitamin C, vitamin D, whatever. As long as you understand that that's what their function is to go into that active site and make that active site even better at breaking those bonds or forming those bonds, that chemical environment is very unique, and those guys are needed to make it. And they can speed up, they can make the reaction like thousands of times faster by doing that. Yeah, so that's it for that. Inhibitors, let's see, what are the two inhibitors that he wants you to know? Good, competitive inhibitors and non-competitive inhibitors. And believe it or not, he's actually not a prick about nomenclature, and I kind of like that about him. So, I said that this was a tertiary protein and that there's an active site. We're going to add just one more thing down here called an allosteric site. So why is that important?
After the conformational change, the allosteric site is the one stimulus.
Good. So we'll mention that later. Let's actually talk about competitive inhibitors first. So what does a competitive inhibitor do?
It competes with other things.
Competes with the substrate at the active site. They're not enzymes, but receptors follow a similar set of rules that enzymes do. They undergo conformational changes. They have unique molecules binding to them. I am disturbingly dependent upon caffeine, like not healthy. okay? The reason I'm so dependent upon caffeine is, well, caffeine's a really good stimulant that wakes you up. The way that it works is, adenosine is the molecule in your neurotransmitter in your brain, and caffeine is structurally very similar to adenosine. So adenosine binds to this receptor and it makes you feel kind of foggy. But if you take caffeine, a caffeine binds to the receptor and then nothing happens. So you're shutting off signals that would normally make you feel kind of tired. And so by consuming lots of caffeine, you feel really good. However, the problem is your body will compensate for that lack of adenosine reception and will produce more adenosine, which is why we say there's like a crash that tends to happen afterwards. But hey, really great up until that point happens. So anything that competes, competitive inhibitors tend to be structurally, chemically similar to the substrate.